actually in the United States. So when I talk with the PI, John Hopkins, Dr. Rothstein, he said that they see good effects of the phase 3. Okay? So we have this compound. Of course, first we have our target. Now we have compound, and we have a model of Huntington disease. We're using mice, type of mice, called R62, which basically they have about 123 CEG repeats. So the protein is all mutated. What you can see compared to this human control, you have reduction of the striatum, a reduction of the cortex, and mainly, if you remember, this is basically glutamatergic projection from the cortex here to the striatum, of course. I mentioned the nucleus accumbens, but the striatum is also part of the nucleus accumbens. Well, nucleus accumbens is part of the striatum. Anyhow, so what we did, I'm going to go briefly. So what we did, we have a model we treated with at week 6, when you can see there is behavioral abnormality, which is including moral dysfunction, and we treated with the compound for 5 weeks, and then you get, of course, the expression of the protein, because what it's going to do, I showed you in the cartoon, the compound will upregulate the transporter, okay, or activate, but mainly, of course, when you activate, you may also, it's associated with upregulation. So what we did, we did also the knowledge flux and some of the behavior. So here is what a way that pharmaceutical industry, what they test compound for looking for any behavioral abnormality. And this is what important class B. So consider this guy, Huntington disease guy, okay? This wild type. So this one is class B. What we see when we give compound, you reduce this class B. Okay? So you have less class B. What does mean? Compound is working well. Can you go back? Okay, I can go back. We did some of the behavior. I'm not gonna go further because of the time. Class A is climbing, the drug improve all this behavior. That basically it's affected in Huntington. Okay? So how it does work? When I put together that cartoon, the hypothesis was there is upregulation, and it's true. When you give compound here, this is even the wild type, you upregulate the transporter, this is in the cortex and striatum. And striatum and cortex, we showed you in the human, those pathological pictures that I showed you in human, there is a reduction of the volume in the striatum and the cortex. So because the cells that are dying, the mutated protein, or maybe barabutyrate, is killing those cells. Okay? So here we look at the cortex, of course, striatum. We look at also other ages. We see good effects with compound. Okay? So I'm going to keep going. Now I'm going to move. So we tested the compound. Now we have a compound targeting at a system that allows to not only upregulate the transporter, but it's also going to help in reducing, attenuating those behavioral abnormality that's seen in Huntington disease. Now the cocaine. So the cocaine, when I was kind of telling you about the alcohol, the cocaine, it shares the same neurocircuitry, okay? But the neurochemistry is kind of different. So they do share the same neurocircuitry, but neurochemistry kind of different, okay? So what I showed you here, for example, for alcohol, we have the cortex, projected in the nucleus accumbens, ventral tegmental area, and then you have your dopamine. Okay? And this is just kind of telling you about the GH1 is right here, and what it's going to do, it's going to kind of regulate the glutamate uptake. Okay? So this is what we use. We use what we call, this is a cocaine model. What we do, so we have a rat, what we do, we kind of introduce the catheter to the vein, the jugular vein, okay? And so we can hold that catheter back in the top here, okay? This way it's easy. When the rat's gonna press in one of the bars, it will you're going to see there is diffusion of the cocaine. Okay? So, but first we train them how to do the food. So either they're going to press this button, this, they will get the food. You train them to seek the food. You kind of show them. Then we do the surgery after. Okay? Then we put them back and see if they are learning very much or not. If they lose with the surgery, they may lose. But most of them, they do good. Okay? So we have a setup here, and we put them. And what they do, the first day, they can press in one of the bars. One of the bars, when they press the bar, there's light and tone associated with cocaine infusion. Okay? So when they press the bar, you have light and also the tone, and that's going to be associated to cocaine. If you remember about Pavlovian or preconditioning. So that's how we are doing it. Okay? So here we call self-administration. This is now we are self-administration, right? That's self-administration. Extension, what we do, we take this wire and we just put them, there is no food, no nothing. So they can keep pressing until there is nothing. So there is no food released. Of course, there is no cocaine. And when they give up, then you can see they are extinguished. So there is no, they know that there is no cocaine, there is no food. Then we put them what they call reinstatement or relapse. That's one of the problems with cocaine, it's relapse. So, you can have, it's very difficult when a patient, when you deprive a patient, well, at least addicted to cocaine or alcohol, when you deprive them, they're going to go back. And that's why we are targeting that phase. So what we did here, we injected the drug during extinction. So when they are giving up, we injected the compound for 5 days, okay, during this exchange phase, and we put them back. See if they're going to see cocaine. Okay? So really, when we put them back, in fact, this is the diagram. This is self-administration. So you can see they press a lot because they want cocaine. Okay? So this is how many active level they are pressing. Then the extinction, basically, they give up because there's no cocaine. They are not interested anymore. Okay? So when we give the compound, here we have 4 groups, of course. One, we're going to inject the vehicle with stadium. The other one is 50 milligrams of cetirizine. And this is, again, during extinction for 5 days. Okay? So we give this compound here at different doses, 50 milligrams, 100 milligrams, 200 milligrams. But what we can see here, you can see there is attenuation of relapse to cocaine behavior. At what dose? At higher dose, 100 and 200. Okay? Lower dose, we didn't see anything. And just bear with me, you're gonna kind of get the sense why the higher dose is working and the lower dose is not working. Okay? By the way, well, before I give you this, when we challenge the rats during self-administration with compound, we didn't see any effect. So we see an effect during just relapse. When you give them the drug during extinction, and then put them back and see if they can see further. Okay? So with 50, we didn't see any upregulation of the transporter. Okay? But with 100, well, at least here 200, we're showing 200, you can see this is basically in a different area of the brain, cortex, and of course this is the accumbens, nucleus accumbens, which is one of the central drug reward, and this one is 50. So when you give the drug, of course it does upregulate the transporter at high dose. So we see good effects with higher dose, but not with lower dose because the higher dose is upregulating the transporter. Okay? So, when we were actually working at this project, somebody else was doing the same thing. So we have to be careful, and especially when you have a good data, be careful of the data. What's happened, we submitted the abstract, and so the result was out, and somebody has a huge lab, just hired 2 people. Well, he had 2 people. They just work in Lighton, and he can replicate the same thing. So we published the paper in 2009, and he published paper in 2010, replicating our data, of course with different doses. But good news, you know, were happy because somebody else certificated our data. But we were kind of have an experience. So if you publish data before, then the quality of the work is going to be. But anyhow, that's what's happening in cocaine. I give you briefly what's happened in Huntington disease and now cocaine. Okay, so I'm going to go very quickly because I have a sign here and talk about alcohol very briefly. Neurocircuitry, it's same, you know better. And we are using a model called alcohol-preferred rats. This is basically what we call a bred rat. They are coming from father and mother, they are heavy drinker, okay? So of course they are the babies or those guys, they're gonna be also developing addiction. So the rat, they drink flour alcohol. What we do, we put them for 5 weeks, okay? Free choice, 15-20%, which is equivalent to whiskey if you heard about it. But it's a higher dose, so you will put them in a free choice of alcohol at different concentration, 15 and 20% for 5 weeks. During that 5 weeks, they develop what they call dependence. Okay? So week 6, what we do, we charge them with compound. Or here we use different concentration, 25, 50, 100, 200. And I'm going to tell you why we went down to 25. You're going to be surprised. So we are targeting again glutamate transport 1. We measured alcohol intake for 15 days, and etc. So what we've seen, I can summarize this, but just bear with me. When we tested different concentrations, 25, 50, and you remember 50, it doesn't work in cocaine. But here we tested 25, 50, 100, 200. All of them did work perfect. Well, not perfect. The only one has a long-lasting effect. Because remember, we treat them for 5 days, and we just kind of continue measuring alcohol intake, okay, for 15 days. But the only, the long-lasting effects we see, it's only with the higher dose, 100, 200. So, and this is, I had the trouble actually convincing the reviewer, well, it's because now you talk about using a transport, but then your lower dose, it's not applicable to transport. So what we do take either it's activating in a different way with transporter, or it's doing something else. You see the pharmacology, how it's very complex, it's really complex. You have to look at everything. Sometimes compound, we talk about non-specific compound. And a lot of them, that's why the side effect. So the compound not only applicate, also doing something else. And that's what we are kind of busy working on. Okay? So I'm going to go, we tested the compound with sucrose. You can ask me because this is very important. It's like a control appetite, see if it's going to kind of have any effect in other liquid. For example, at this point we use sucrose. We didn't see any effect. Okay? So this is kind of a Western blot that I showed you cocaine and Huntington. So really, the higher dose, you see effects, 50 and 50. You don't see upregulation, but you see effect. And I really, until now, I don't know why we see effects. Compound is doing many things. It's not only upregulating the transport component, but the good news, long-lasting effects, it's you see long-lasting effects with higher dose of the drug. So I think this is another area. The summary, what we see in the compound is working in Huntington disease to upregulate glutamate transport 1, cocaine, alcohol, etc. And this is basically the group that's happening. Without them, I'm not going to present anything. And all this work, as I mentioned, is supported by the National Institute of Health. Basically, it's R1 that's supporting this project. So I would like to take all these people, and I would be happy to take any questions.
Thank you very much.
All right.
Thank you very much. Quite interesting and confusing at the same time.
Confusing.
Okay. Yeah. So I would like to say, do you recommend safe for treatment of the Ash disease?
What is it?
Huntington disease. Would you recommend this drug for treatment?
I tell you, this is a good question. I've been asked several. Each time I give this talk, I've been asked. Well, I recommend it. Yes, I recommend ceftriaxone for treating, but I would recommend combination.
Okay. For how long? For how long time factor?
This can be whatever.
Any adverse effects so far concerning the moral in terms of depletion and the susceptibility of the patient to the infection?
This is a good question. Like the ELS, they tested actually long-term effective compound, and it seems like there's no toxicity. So when it's in phase 3, treating, so we talk about hunting, the ELS is also a chronic disease. So they are tested it in ELS, and it seems like it's working very well.
So you are suggesting that the next era will be witness the emergence of the antibiotics for treatment of the neurological disorder?
Actually, there is study, there's a lot of people, if you type ceftriaxone and brain or whatever, you will see there is study in ischemia. For example, you have a patient. Well, ischemia is kind of very tricky. You have to apply the drug right away. When you, for example, if somebody has stroke, you have to give it right away. So it's kind of very tricky. But there is study in animal model testing ceftriaxone and stroke ischemia. What else? ELS, of course. You talk about ELS and T10 disease.
Thank you.
Okay, thank you, Dr. Yusuf again. Thank you very much. Our fourth speaker in this session is Dr. Bashir Jarrah. He is a full professor in histology and histochemistry in Al Jouf University of Saudi Arabia. He is the author of 70 papers in scientific journals and the author of about 25 books in the field of histology and environmental histochemistry. His talk today is hepatic histological alteration induced by sildenafil overdoses. So welcome with me, Dr. Bashir.
Bismillahirrahmanirrahim. I'm happy and pleased to be with you. And in fact, I feel jealous because most of you are coming from very famous research institutions. The title of my topic is the hepatic histological alteration induced by sildenafil overdoses. This work was carried out in the Department of Medical Laboratory Sciences, College of Applied Medical Sciences at Al Jouf University. First, I would like to introduce my university. Al Jouf University is a new one. It just started 9 years before. Now we have 14 colleges. Pharmacy is one of the pharmacy college is one of them, and with the total students, almost about 22,000. Now, in my presentation, I have 3 domains: the sildenafil, Viagra, and the overdose, and the third one is the histological alteration. Now, as you know, sildenafil acts as a selective inhibitor of phosphodiesterase 5 that causes a release of nitric oxide from the benign nerve ending, leading to the relaxation of the smooth muscles of the penis, increasing the inflow of the blood into the erectile tissue, and by then causing an erection. Now, sildenafil overdoses uptake is mainly seen among men suffering from ED, erectile dysfunction, where the stigma of this disease surrounds them, especially with their parents. The Viagra or sildenafil is an effective drug in elderly men, but had a lower efficacy rate with increasing age, and here there is a space or a chance for the overdose. Also, this drug is popular among young people. So sildenafil mortality is increasing with young adults due to at least for the following. First, there is a belief that it increases libido, also, or it improves sexual performance. The third, increases penis size. Also, here there is a space for the overdose. Also, this drug is used among athletes and the people who are included in sports. Research studies also indicate that sildenafil is widely used by bodybuilders and athletes, and so far is legal in the world of sports. Sildenafil pumps more blood in their muscles. Also, Viagra is mixed with good plants in different countries, especially in Lebanon and other countries, and South America. And most of these products contain sildenafil and are available on the roadside. So there are many spaces for using overdose of sildenafil. Case studies showed that sildenafil overdoses may result in facial flushing, hearing impairment, nose stuffness, nose bleeding, hypertension, chest pain, painful breathing. Sometimes it's up to 24 hours duration, tachycardia, and arrhythmia. Sildenafil overdoses was reported to be fatal also due to its arrhythmogenic potential. The fatality rate is on rise and have reached crisis levels in certain countries. The first Viagra-related death was reported in Singapore in 1999, and the last case, this is my prediction, the last case, the study was reported from Jeddah, Saudi Arabia, where a Egyptian man passed away while he was in the airport waiting his wife in the Arabic section. The forensic report indicated that he took 4 blue pill. Blue pill is the other name for the sildenafil, and had a history of, and also they found that he had a history of heart disease. So we started the work 2 years before in my laboratory, and I started to work about the histological alteration induced in the testicular tissue. We exposed rabbits to overdoses of sildenafil, and we found that it had provoked a tubular and interstitial histological alterations. Mainly, our finding that spermatocytes carry prognosis. Second, spermatocytes degeneration, and third, and which is the most important, is the spermatogenic arrest. I'll show you some these here. What you found, in fact, is enlargement in the testes, not something else. All rabbits that received overdoses, they showed enlargement of the testes. And this is the comparison between the testes of treated rabbit with the control rabbit. You can see the difference in terms of size. Now, this enlargement, it was, as a matter of fact, as a result of the congestions of the testes. You look at the blood vessels here, and the interstitial tissues, it's almost completely congested. Besides this, if you compare, we found that these are the surface tubules of the control rabbit with the treated one. Here, everything is normal. We find the stages of spermatogenesis, starting from spermatogonia, from spermatocytes, spermatocytes, spermatids up to the spermatozoas, which are seen in the lumen of the tubules. Here, almost the tubules are damaged. We find despermatization, mainly spermatids, but not spermatozoa in the lumen. Here, this is a normal tubules of the control, while here, and this one, this is the tubules of a treated rabbit. Here we found no spermatozoans at all, just we found host cells. And this means that almost there was a complete spermatogenesis arrest. This work, we published this work in the Journal of Metabolic Drugs and Toxicology, and a few months ago, almost before 10 months, I started to work about the histological alterations that are induced in the liver. We found in literature that there's indication that liver plays an important role in the detoxification of sildenafil, where the bioavailability of this drug is determined by the pre-systemic first-pass hepatic metabolism. Also, I learned that the histological and the histological characterization in the tissues due to sildenafil overdoses intoxication was not being well identified. So I started the work. So the objective of this work was to characterize the possible surgical and histochemical changes following experimental sildenafil overdosing. 30 male rabbits, almost the weight of 1 kilogram, were used in the present study. The rabbits were randomly assigned into four or five groups. Control one with a number of 6 rabbits, and other 4 test groups of 6 rabbits each were separately caged. Concerning the dose, sildenafil tablets containing sildenafil citrate equivalent to 50 milligram of sildenafil were used in the present study. Members of the treated groups were exposed to intraperitoneal injections with a daily single dose of sildenafil. The first treated group received 1 milligram per kilogram, the second 3 milligram per kilogram, the third one 6, while the last one 9 milligram per kilogram, by the way, for 35 days. After that, fresh portion of the medium group of the liver from each rabbit were cut rapidly, were fixed in 10% natural buffer formalin, and then dehydrated with ascending grades of ethanol, 70%, 80%, 90%, and 2 changes of absolute ethanol, by then treated and 2 changes of chloroform before being impregnated with 2 changes of methyl paraffin wax and embedded and blocked out for preparation of the histological sections. Histological sections were prepared with a thickness of 4 to 5 micrometers, and they were stained with the following stains. The hepatoxylin and insulin stain, this is a routine one for the general structures of the tissue. Periodic acid shift stain, this is used usually to detect the change in the glycogen or neutral mucous substances. The third one, malonic trichrome, it is used to detect the situation of the collagen fibers and crucial two reactions, or it was used to detect if any change in the hemoglobin pigmentation. Our results were as follows. In comparison with the control group, the following histological and histochemical alterations were detected in the liver of sildenafil-treated rabbits. Concerning the hepatocytes, mainly, the nuclei of the hepatocytes, we find the following alterations: nuclear vesiculation, anisnucleases, binucleation, carbonucleases, carboxylases, and cariolysis. Here in this diagram, I can, for example, this is binucleation. Here you have carbognosis. Here we have carboxylases. Here we have cariolysis. So we find different pattern of alteration in the nuclei of the hepatocytes. What does each mean from these alterations? Anisnucleations, which means that different sizes of the nuclei of the hepatocytes is known to be associated with the hepatic dysplasia. While binucleation represents a consequence of cell injury, usually seen in regeneration cells, and may indicate nuclear interruption in the mechanism of sildenafil detoxification. On the other hand, carboxylases is an irreversible condensation of chromatin in the nucleus of the cell undergoing necrosis or apoptosis. The other nuclear changes were the carboxylases and cariolysis. These are destructive fragmentation and complete dissolution of the chromatin matter of a nucleated or dying cell. The other alterations we found that most of the hepatocytes were nucleatics. Significant necrosis was noticed in the hepatocytes of sildenafil-treated rabbits. This alteration was more pronounced within the periportal zones, the hepatocytes, where some of these cells stained more blue than normal. Now, this figure shows that almost most of the hepatocytes, they have necrosis in their cytoplasm. Almost most of the hepatocytes are nucleated here. what's the indication of necrosis here? As a result that the seen hepatocytes necrosis might indicate oxidative stress on these cells as a result of sildenafil toxicity. One more alteration is about the hydrogen degeneration. Hydrogen degeneration, which is a cloudy swelling cytoplasm, was seen in the sildenafil treated rabbits. This alteration increased in the severity with increasing the dose and duration of sildenafil exposure and associated with necrosis. Here, if you see most of the cells, they have almost transparent cytoplasm as a result of the replacement of water for the organisms and the contents of the cytoplasm. Almost most of the cells, they show this alteration. Now, what does it mean, hydrogen degeneration of the hepatocytes? It might be a result of ion and fluid hemostasis disturbance that lead to an increase of intracellular water. Also, this change or this alternation might be exhibited as a result of disturbance of membrane functions. So there is a change in the entrance of water to the cells of hepatocytes due to the sildenafil toxicity that lead to massive influx of water and sodium ions due to the toxicity of this drug. Now, one more alteration, we found that there was a bile duct hyperplasia. The dilation of the bile duct was seen together with the proliferation of the lining epithelium in the treated rabbits in comparison with the control ones. Here, this is the bile duct hyperplasia. It's very clear. What does it mean? Now, the seen bile duct hyperplasia may indicate that sildenafil and or its metabolites are excreted via bile secretions with an irritation effect on the bile duct epithelium. One more, it's the inflammatory cell infiltration. An aggregation of inflammatory cells infiltration mainly in the hepatic portal space was seen. The infiltrated cells were mainly lymphocytes and the plasma cells. This is the infiltration. It's very obvious. Most of these cells are lymphocytes and plasma cells. And this suggests that the drug interacts with the hepatic interstitial tissue, interfering with the antioxidant defense mechanism and leading to reactive oxygen species production. Maybe this is the last alteration. Kupffer cells have hyperplasia. The sinusoidal Kupffer cells became prominent and increases and the number to the exposure. It's not only increases the number, but there was also an enlargement in the Kupffer cells size. This means that the induced Kupffer cells hyperplasia might be a result of increased autophagy throughout the hepatic tissue to help in removing the accumulated drug and its metabolites. Also, this alteration might be correlated with the amount of hepatic tissue injured induced by this drug. One more. Also, we found that there was a congestion of hepatic vessels. As you see here, the blood vessels, the small ones, and even the central one, are congested. Occasional congestion of portal vessels and the central veins were observed. This could be resulted from the injury of hepatic blood vessels by the drug. We have slight portal space fibrosis. I'll run to the picture here. This is the fibrosis of the portal space. And we have also partial glycogen depletions. Past stain showed that evidence of partial glycogen depletion and heterogeneous pattern in the hepatocytes of sildenafil-treated rabbits in comparison with the control ones. The depletion was more prominent in the liver of rabbits exposed to 9 mg/kg for 35 days. Now this is a comparison. This is the normal one, and this is the treated one, and you can find the difference. It's very obvious. The induced depletion of glycogen storage by this drug may indicate an effect in glucose absorption or in the enzymes involved in the process of glycogenesis or glycolysis. Microbilation from this study, one may conclude that the chronic exposure to sildenafil overdoses produced significant histological and histochemical alterations that might affect the liver function as a result of hepatocytes insultation. These findings might also indicate a need to study the effect of sildenafil among patients with hepatic impairment and to test the safety of using sildenafil among elderly people where liver functions are significantly decreased. Thank you.
Thank you, Dr. Bashir.
Thank you very much. I have two questions. First, why you choose the liver rather than the testis itself when you saw a lot of changes in the testis in terms of degeneration and circulatory syndrome? That's the first question. Yeah.
Now, as a matter of fact, we found that there is drastic changes in the testes, and we want to test the other vital organs, the liver and the kidneys and maybe other organs.
Let me rephrase it. Why you didn't study the testicular changes? Why these testicular happen? I mean, is there an interpretation and an explanation why these testicular changes happen with this drug? Do you have an explanation?
I think it might be as a result of pumping a large amount of blood in the pelvic area.
And that will be good nourishment.
Yeah.
So why did you
No, but it is
a lot of oxygen or of vitamin?
no, but it's more than needed because most of the blood is pumped towards these organs, the pelvis area, and the tissues are enlarged. It is not normal.
Okay. The other things, in terms of the liver, are those changes that you described in terms of the hydrodegeneration till necrosis, are these changes seen in all animals or those at different intervals?
No, not all. Now the changes or the alterations we have seen more duration and more concentration. So in the last group that received 9 milligram per kilogram, these alterations were more prominent. While the first group, which received 1 milligram per kilogram, which is almost normal, the normal one, the normal dose, we saw very few alterations.
But I didn't see any images of the control and the test animal at the same time to see if there's an inflammatory changes in the liver. You talk about the inflammatory changes in the liver. I would like very much to see the control one, if there's the same thing.
I have the controls, but there is no space for
Okay.
Victor, was all the time to get me to finish.
Thank you.
In fact, I know.
Thank
15
you.
minutes is not enough.
Thank you.
We'll take one question, please, because we are running out of time.
Thank you, Mr. Mixture. First, this guy in Riyadh airport have died because he had overdosing. But of course not because of the liver, because this is acute syndrome, something acute. Second, if we take, the question is, the metabolites of sildenafil, are they as active as sildenafil or inactive or?
No, I'm not pharmacologist, but I know that there are four metabolites. One of them is very active. The metabolites, they are active. This one, there are four metabolites.
Active as sildenafil itself.
I'm not sure of this, but I know one of them is very active as sildenafil itself. One of them. One of the four metabolites.
What do you think about having the formulation of sildenafil as, for example, bioadhesive bunker carrier to avoid bypass metabolism in the liver, to avoid these harmful effects of the liver?
Well, doctor, I can't answer the question.
If we formulate sildenafil in bunker between the cheek and the gum, put it with bioadhesive polymer and put it, we do this in pharmaceutics, okay? And then the bypass to the liver is avoided this way because it goes directly to the circulation without passing to the liver. What do you think of this? To avoid
I don't know, but I know that the site of detoxification for the sildenafil is the liver. This is not in the literature.
So if we avoid the liver,
I can't tell about this. I can't tell.
yeah. But how will these very dangerous effects effects are there, and this drug is widespread and everywhere.
Yeah,
Majority of people are taking
I
it.
started that and I'm talking about overdose.
So Pfizer
These are alterations used by the overdoses of stability.
I mean, when having a NDA for this drug, new drug application for the FDA, how it comes that this company have succeeded in having the approval, marketing, authorization for such a
I have seen the analysis of the company, and most of them they are biochemical analysis, and with the normal normal dose, and most of the time it's sufficient results.
What a normal dose for would be?
It's 50 to 100 milligram per
50
daily, which means almost 1 milligram per kilogram.
Not every day, every week, for example.
Yeah, at once, huh?
Anyway, thank you, Dr. Bashir, again. Thank you very much. Our final speaker for this session is Dr. Sausan Abu Hamdi. She has her PhD in pharmacology from Durham University, UK, in 2007, and a postdoc in pharmacology also from Granada University in 2008. She is currently an assistant professor at the Department of Biopharmaceutics and Clinical Sciences at Jordan University. Her talk today is Potential of Melissa officinalis essential oil in agitation and anxiety. So please welcome Dr. Sausan.
Okay, my specialty is in neuropharmacology, and to link it with the title that I'm going to talk about, Melissa, we could say in neuroscience that Alzheimer's and dementia are the major serious neurodegenerative disease affecting the individual and a lot of old people. Now, usually the physician treat this with attention directly to decline in the cognition decline. But physicians with the treatment, they focus on many people with the late stages of disease. They suffer with the complication stage. It's a type of behavior and psychological symptoms that is very distressing to the patient and to the caregiver and to family. And the solution for this, that usually physicians, they prescribe antipsychotic agent to decrease the complication that usually associated with the disease. Now, many old people, they have poor toleration for this medication and usually associated with multiple adverse effects, such as, for example, it lets the patient feel drowsiness, decrease in awareness of the well-being of the patient. There is a high incidence of falling. That means we need alternative. And a new target for alternative therapy has been identified or linked nowadays in research and using herbal medicinal plants to overcome the complication in my presentation like agitation and anxiety. One of the plants that has been identified to be successful in this field is Melissa officinalis. We can say that many previous studies have been identified the usefulness in this plant in the control or treatment of agitation for patients with the late stage of Alzheimer's disease in many randomized trials. But the pharmacological data is limited about the mechanism of action of this essential oil that will have benefit in the patient treated with this type of oil. So our aim was in our study, let's find the pharmacological effect of this plant. How it will be useful for treatment of late stages of Alzheimer's or dementia. So in neuropharmacology, we use a technique known as patch clamping technique. And in this, we use a primary cell culture neuronal cells from here. We could